Hello everyone. Today we're going to talk about the roles of computers in drug discovery. So this is the fourth week of our computational chemistry class. So let's get started. So today, we're going to talk about, how much computers are playing important roles in drug discovery in the modern days. So pharmaceutical industry can be our next economic boost. So our government is trying to push this direction so hard. So if you look at the numbers, the top 10 pharmaceutical companies all around the world, all around the globe, they made about $440 billion in 2017. However, Korean companies still, occupy less than 1% in the global market. So for example, there is a very large companies called like Roche or Pfizer, AbbVie, Johnson & Johnson, and Sanofi, Merck, Novartis, and Gilead, and So there are many global multinational pharmaceutical companies, and they are making huge amount of money. Recently, so there are some number of trials in Korea too, to develop new drugs. So recently, Yuhan succeeded in developing a new drug. Actually, it's not a completely new, but it's a improved drug to treat certain kinds of cancer. So as you can see, there is an article, which was published in 2018. So Yuhan sold their molecule to a big pharmaceutical company, to Janssen, by $1.25 billion. A new drug they developed named Lasertinib. So the Lasertinib is a new drug that works for lung cancer patients with drug resistance with existing, lung cancer drugs. And they also identify that this new drug, Lasertinib, works for patients who have cancer cells in brain. So the certain type of lung cancers are known to, transfer to brain and which is very lethal, which is very, critical. And it is also known that the target of this drug, so the target means that a protein that, as we talked about, we talked about this in the previous class, right? So the target means that a specific protein that this new drug binds. So for Lasertinib, it is known that a target protein is EGFR. So what is EGFR? So the EGFR is a very, famous, one of the very famous targets in biochemistry. So the EGFR is an abbreviation. So it's a abbreviation of epidermal growth factor receptor. So the epidermal growth factor receptor means that, so they control the growth. They control especially the expression of proteins and growth of cells, etc. So it means that they transmit signals to cells. So usually, EGFRs are located at the at cell membrane, and as you know, most of the biological signals happening in a cell are actually chemicals. And this is the structure of one of the EGFR. So there are many kinds of EGFR in human body, in human cells, and you can see the structure of this protein with this PDB ID. So usually EGFRs are stuck at the protein at cell surface, cell membrane, and once they bind with a specific ligand, then they transmit signals So in biology, usually the signals are transmitted through the phosphorylation, right? Phosphorylation. So phosphorylation means, so enzymes attach phosphate group. Phosphate group means PO4, right? I mean, so this phosphate group, they bind phosphate group to specific proteins. And these phosphorylation causes the cascade of signal. Cascade means, the transmission of the signal. So in summary, so if a ligand binds to EGFR at cell surface, then it forms a dimer. Dimer means two EGFRs bind to each other, and phosphorylation of C-terminal tyrosine occurs. And then signal, goes along to the, goes along through the many, proteins, and it goes through the until the nucleus of a cell. And this signaling pathway turns on and initiates. Initiates means it starts cell proliferation. So the meaning of proliferation is written here. So they control, how cells respond to the, external stimuli, external, signals. But the overexpression of EGFR usually leads to cancer. It is known that the overexpression of EGFR leads to cancer. So some type of cancers can be treated by blocking, these, overexpressed EGFRs. Then how do anti-cancer drugs work for, EGFR? So the phosphorylation occurs by transferring a phosphate group from ATP to tyrosine. So the source of phosphate group is ATP, and the phosphate groups are transferred from ATP to the tyrosine of EGFR. Then how does, a lasertinib works? And lasertinib and similar EGFR binding cancer drugs actually bind to the ATP binding pocket, binding site of EGFR. So if you take a look at the example, so we talked about the PDB database, right? So if you go to the PDB database, I recommend you to, do it by yourself. So if you go to the PDB database and if you search, the protein with an ID 4I24, then you can see this structure. So as you can see that this is the, description of the structure. So the title says that the structure of T790M EGFR kinase domain co-crystallized with dacomitinib. So this is another, anti-cancer drug. So which means that, usually, the ATP is bound here. ATP is bound in this pocket. But when this kind of anti-cancer drug comes in, then ATP does not bind here. Instead, this, drug binds here, and it, prevents ATP from binding to this pocket. Okay? So this is how generally, drugs work. So they bind with protein instead of, natural ligands. Then what was so special about that, new drug, lasertinib? So I'll just, tell you a little bit more detail about this drug. So if a patient has the T790M mutation, so the 790 mutation means that so in biology, in biochemistry, so this, T790M means that threonine at 390 position. So which means that, 790's residue is usually threonine. But sometimes it can be mutated to methionine, okay? So if you, think about what you learned from the biochemistry class, a protein is a sequence, is a chain of 20 amino acids, right? And threonine and methionine, they both are amino acids. So usually, so the 790th position should be occupied by threonine. But sometimes if mutation happens, mutation means amino acid is changed from one to another, right? So that if the mutation happens from threonine to methionine, then most, known lung cancer drugs do not work, because most known lung cancer drugs are designed to work with the protein, with the wild type protein, which has, threonine at 790th position, right? And unfortunately, it is also known that, about 40% of lung cancer patients have T790M mutation. Okay? So, So which means that 40% of lung cancer patients don't have any, drugs. If they have, this kind of mutations. And before that, and for those cases, actually, there was only one drug, which can treat, such, mutated cases. It's called osimertinib. So osimertinib was a known, lung cancer drug with this specific type of mutation. And what, people in Wuhan did was that, so they changed it a little bit, so starting from this, osimertinib, they changed it, this group, and if you compare these two structures, right, so you can see that, this group is a little bit changed, and this group is also a little bit changed, right? So by changing these two, and they made it penetrate the barrier called blood-brain barrier. So the blood-brain barrier is actually, so in our brain, actually our brain is, surrounded with a barrier. So it's a membrane, and this blood-brain barrier protects brain from infection, right? It protects brain from viruses and some other toxic chemicals, and so it's very hard to penetrate. But as I told you, so some type of lung cancers are transferred, to brain, so which makes it very dangerous, right? So, but unfortunately, osimertinib was not able to, penetrate. Was not able to go into your brain and treat, those tumors. But after changing these groups into these groups like this lazertinib appears to be able to, penetrate this blood-brain barrier, and it seems that it can work for tumors inside the brain. So it was shown that, it can be used to treat patients whose cancer cells are metastasized to brain. So which means that, tumors are, transferred, the patients whose tumor cells, are transferred to their brain. Okay. So this is what, usually, drug companies do. So they start from some known drug, and they make some changes. They make some changes in some functional groups, and they make it better. So that they have, desirable properties. So here, we have a concept of, the concept of permeability comes in. So let's talk briefly about membrane permeability. So mostly, as I told you, as we all know, so cells are actually, if let's say that this is a cell, and cells are actually, usually surrounded with cell membranes, okay? cell membranes. And cell membranes actually consists of this kind of lipid structure, this kind of structure. This is the bilayer, lipid bilayer. It's called lipid bilayer because it consists of, two layers of lipids with an opposite direction, okay? Oops, sorry. Yeah, with an opposite direction. So, and for one drug, actually, so let's say that this is a drug, and for one drug to work, then it should penetrate the cell membrane. It should penetrate a cell membrane, and it should go into the inside the cell, and it should bind with a specific protein, okay? So to go through a lipid bilayer, a drug should not be too hydrophobic or too hydrophilic, nor too hydrophobic. Because as we all know, so let's say that this is a lipid molecule, then lipid molecules actually generally, these head groups are hydrophilic, right? But these tail groups are generally hydrophobic, okay? So, if a drug is too hydrophilic, then it will just stay outside or inside the cell and it will not penetrate the cell membrane, okay? But if a drug is too hydrophobic, then it goes into a membrane and it'll just, stay inside. Because, hydrophobics like hydrophobics and hydrophilics like hydrophilics, okay? So, generally, drugs should not be too hydrophilic nor too hydrophobic, okay? And so far, in most of the cases, for drug discovery or for drug design, the human chemical intuitions played an important role. So to control the, these kind of, hydrophobicity or other kinds of, to change other kinds of properties of molecules. So let's briefly so we've talked about some, real example of drug discovery. So let's talk about a brief and general overview of drug discovery. So let's review the drug discovery and development process. So actually, drug discovery and development process consists of multiple steps, many steps. So in this picture, so they are showing, multiple steps of drug discovery. So the first, we should start from target identification, okay? So the target identification means that, so what protein causes, disease, causes a disease. So we should know, so what is our target protein, okay? Oops. And after that, so we should do target validation. So we should validate whether this is really our target or not. And we should do a target validation. And after that, we should find the hit. We should find the hit molecule. So the hit molecule means that they are small but possible candidates that can be drugs. So the hit is the very first step of drug discovery. But usually, so these hit molecules can be toxic and they are generally somewhat small. But starting from a hit molecule, they improve it to a lead molecule. So the lead molecule is so usually it's called like this in Korean. So the lead molecule is the base structure to develop drugs, okay? And from lead molecule, we should optimize it. So the optimize means, so we make it better. So after, doing some experiment, we analyze data and, we consider, protein structure, and people think that, if we change this group into another group, then, the drug will be more, efficient, or will bind with the protein more strongly, okay? So once they do it, and after that, they go to a, preclinical step. So in preclinical step, usually they check toxicity by lab test and animal test. So in the preclinical test, so they see, they check, whether they really work, and check whether they are toxic or not. And here, so if it is identified to be non-toxic, so it is okay, so it is not, harmful, then after that we go to a clinical trial steps. So the clinical trial means human experiment. Okay. So, So if we talk about this, again, so this is the drug discovery, steps. So first, so we should start from the target identification, and after that, so we validate our target, and usually, so we use, for this step, usually people use, knockout mouse. So the knockout mouse means that, some mouses without, certain genes. And after that, so we find, hit molecule, and from hit, so we change it to a lead molecule, and after that, so we do a lead from lead, so we do lead optimization, and after that, so we find, drug candidates. So once we find a good once we found, good drug candidates, then we start a preclinical step. As I told you, so during the preclinical step, we usually check the toxicity and ADME. So I'll talk about ADME later. And once it is proven to be non-toxic, then we go to a clinical trial. Clinical trial means human experiment, and usually, clinical trials are separated into 3 phases. And once they pass all these 3 phases, then a new drug is approved. Okay, then let's take a look, what kind of, phases are there during the clinical trial. So during the first phase, so usually, toxicity and side effects and dosages are tested. So during the phase 1, usually, so it is done with, 20 to 80 healthy volunteers. And the success rate of the phase 1 is about 70%. So during the phase 1, so usually, so they recruit healthy volunteers, and they, dose small amount of drug, and they increase it little by little and see, whether it has some side effects. So if it is proven that there is no side effect, or it is non-toxic, then it passes to the second phase. So in the second phase, usually test is done with about 500 patients. They are real patients. Then they give this, drug candidate to real patients, and they see that whether, this drug is really effective or not. Is it really effective or not? And also they check, side effects. So which means that, so they compare, their effects. They compare the effect against a dummy treatment. Usually it's called, placebo, right? So it's It's a false drug. So whether it's just the psychological or whether, so in other words, it means that they test, whether it's just psychological or whether it is really, useful, really, working. And phase 3, so once it is, so the success rate of phase 2 is about 30%, okay? So which means that, lots of drug candidates are actually failed during the first 2 phases. So, and after phase 2 is passed, then they go to a larger group of people. So up to thousands of people. So it's a phase 3. So usually they recruit 300 to 3,000 patients over many countries, over many ethnic groups, and they check whether the drug candidate is really effective. So, and the success rate at this step is about 35%, right? So if you see, success rates, if you multiply these success rates, actually the final success rate is very, very low, okay? But once you pass this, phase 3 and it is identified to be really effective, then it is proved by, FDA and other government, offices, and they are actually, licensed and they go to market, okay? So this whole procedure takes about 10 to 15 years usually. And they cost a lot because, you should test with, lots of patients. You should recruit, lots of patients and do tests on them. So it requires lots of money. So the drug discovery, process is very, very, thorough and very rigorous, procedure. So let's take a look at some examples, drug discovery efforts happening in Korea, in South Korea. So this is the picture that I adapted from, Hanmi Pharmaceutical Company. And they say that, they are testing, this number of drugs, okay? So as you can see, so this is the preclinical, okay? So this is the preclinical phase 1, phase 2, phase 3, and I explain you, what do they do, so during those phases, okay? But as I mentioned, so during the, phase 1, so about 30% of these may fail. And from here, there are 1, 2, 3, 4, 5, drug candidates, but only 30% of them will succeed. And here, they have 1, 2, 3, and also here, only 30% of them will succeed, okay? So maybe, based on probability, only, one of these will be approved at the end, right? So there are some companies, who are, actively developing drugs, but drug discovery process is quite, risky and very time consuming. So let's say that, you are lucky enough, and once you, got an approval from FDA, then will you make, lots of money? But unfortunately, it's not always true because so far, so there are 29 new drugs, developed by Korean companies. So these are some of the lists, but only 5 of them made more than $100 million. $100 million. So the reason for low market share is that because, there are usually, so better drugs in general. So let's say that, you developed a drug, working for some diseases, but unfortunately, if there is a, better drug, then people usually want to use it, right? then if your drug is not better than the other, then you have to sell it very cheap, right? Which means that you don't make that much money. Okay, so the overcoming existing drug is very difficult, and becoming the best drug for certain type of disease is generally called as the best in class. Okay, so usually the best in class means that the best drug for some certain disease. There's also another similar concept called first in class. So the first in class means that if there is no drug for some certain diseases, then if a molecule becomes the first drug to treat some certain diseases, then it becomes the first in class. So this type of first in class drugs are generally blockbusters, because the first in class means that before that there was no cure, there was no drug. But if your drug is the first in the market, then you can make you will probably make a huge amount of money. But developing these first in class or best in class drugs are generally quite difficult. Then so here is where computer comes in. So why is CADD? CADD means computer aided drug discovery or drug design. And why is CADD important? So actually in computer science field, in actually the computer hardware field, there's a law called Moore's law. So the Moore is from the name of Gordon Moore, who is the founder of Intel. And Moore said the number of transistors in a dense circuit doubles every 2 years. So which means that our CPUs become, twice denser every year. So if you see this plot, so the x-axis is the year and the y-axis is the number of transistors in the CPUs. Then as you can see, so Moore's law actually, is obeyed quite nicely. So every 2 years, so the number of transistors are almost doubled from 1971 to 2016. Actually, so up to quite recently. But the opposite is true in the pharmaceutical industry. So some people call it as the EROM's law. So the EROM's law means that so they just reverted the Moore's law. So if you write down Moore in an opposite direction, reversely, then it becomes EROM. So because as time goes, the number of drugs, the amount of money to invest to drug a new drug becomes more and more expensive. So here so this plot shows the here the y-axis is the number of drugs per billion US dollar spending, US dollar R&D spending, and the x-axis is the year. So if you see this plot, so as time goes, so the number of drugs per $1 billion spending, R&D spending, is decreasing. So which means that in 1950s and 60s, if you spend $1 billion, then you could have developed about more than 10 new drugs. But these days, so even if you invest, $1 billion, so still, so it is hard to develop it is hard to develop one drug. So the drug discovery. So this means that drug discovery is becoming harder and harder and also more expensive. So in order to solve this problem, people are trying to use computers quite actively and quite widely. So here, so in this picture, so this picture shows, what kind of, things computers can do, can help during the drug discovery process, okay? So first, so during the, target identification step, so we can do, sequence analysis, fold recognition, and homology modeling. And for target validation, so we could, check target druggability or tool compound design. And during the lead compound discovery and lead optimization, and so we can do docking and de novo design and QSAR and so on. And after that, so we can do, in silico predictions. So usually, so here the in silico means, so using computers. Because, computer chips are made from silicon, right? So during this lecture, actually, so during this semester, so among these tasks, so we'll practice protein homology modeling and protein ligand docking and scoring, and also we'll run some, example protein dynamics simulations. So let's briefly introduce you about the concept of these tasks. So first, so we'll talk about, protein homology modeling. So what is protein homology modeling? So as I mentioned, so as you must have learned from biochemistry class, so the protein sequence actually determines its structure. So which means that once you have a sequence of amino acids, then its 3D structure is determined uniquely, okay? And structure of many proteins, but the 3D structure of many proteins are unknown so far. So then, if the 3D structure of a protein is not known, then what can we do? Then we can, model its structure. So let's take a look at this example. So let's say that so we know the sequence of this protein, and also we know the 3D structure of this protein. But the protein we want to handle has this kind of sequence, but let's say that so its structure is not known. It's not known. Then what can we do? So then we can model it. Okay, we can generate its structure using computer. Then how can it generate a computer? How can it generate the structure using computers? So one of the most commonly used method is called homology modeling. Okay, so the homology modeling is to model an unknown protein structure based on known protein structures with similar sequences. Okay, so let's say that so these two sequences are similar. So here our main hypothesis is that if sequences are similar, then 3D structures must be similar. Okay, so let's say that so your sequence, you only have a sequence and you don't know its structure. Then what you do is that so you go to the PDB database and you search a similar sequence from the database. Okay, then luckily you found one structure. Then you think that so this sequence is very similar to mine, and I know this 3D structure, so I assume that the structure of my protein, the structure of this blue sequence will be similar to this one. Okay, so I start from this structure and I model the 3D structure of my sequence based on this. So that's what we called, in other words, called template based. Okay, so the template means this. Okay, so we have a some answer. So I mean, not an exact answer but similar answer. So we know the structure of the similar sequence, and from that so we make the new structure. Okay, so that's template based modeling, also known as homology modeling. So the homology means this. Okay, and the other task we're going to practice during this semester is called protein ligand docking. So usually the function of proteins are controlled by docking, which means binding with ligands. Okay, so let's say that this is your protein, and usually this is a small molecule. This is a small molecule, and small molecule binds to this protein tightly, then it changes the function of protein. And docking calculation predicts then how do they bind. So let's say that this is our protein, and we want to know how this one binds to here. So this is the one of the example of docking simulations. So ligands may bind here, or ligands may bind here. Okay, so this is called binding pose. So how do they bind? And also secondly, so we can docking simulation can identify how strongly they bind. So in other words, it is called binding affinity. So usually drugs are strong binders, right? So it's no surprise, because in order for it to control the function of protein, at least so it should bind quite tightly. It must bind tightly with the protein. Okay, right? So that's why usually drugs are strong binders. And the last example is protein dynamics. So at the end of this semester, so we are going to practice molecular dynamics simulation, in short called MD simulation. So in MD simulation, we simulate how proteins move. Okay, so as I told you in the first class, so even with the state-of-the-art experimental technique, so it is impossible to observe the movement of proteins with atomic details. So MD simulations can calculate binding affinities, and also it can show us the movement of proteins quite accurately. Okay, so this is the example of the motion of some proteins. So each line represents the various movements of proteins. So I'll show you some example. So this is the simulation that was run by myself. So for example, so here this is the MD simulation of a protein called adenylate kinase. Adenylate kinase is mediating the chemical reaction between ADP and ATP, and this one is here. The protein is being closed, And in this example, a protein is starting from the closed form and it opens up. Okay, so using the MD simulations, so we can observe this kind of protein dynamics movements. So during the practice session, so we're going to run some of these, examples, and you'll observe, how proteins move. Okay, so far, so we have talked about the, role of computers in drug discovery process, and we will practice, some of those, tasks. So let's call it a day, and we'll do some. So they're going to be a practice session on Friday. So I'll give you a further notice. Okay, thank you.